Welcome everyone to today's AI4growth session. My name is Michael Rademacher. Today we have 4 interesting speakers for you. The setup is as usual. We will first have a little video presentation by the speakers. We'll have a short interview. And at the end of the 4 interviews, let's say around 10 to 5, we will have a live Q&A as well. And that means we're now ready to start with our first introduction, our first use case. It is brought to us by Omina Technologies, Bas Jansen, and we will be watching the video in a moment.
Good day everyone, my name is Bas Jansen. I'm the head of life sciences at Omina Technologies, and today I want to talk to you about inflammatory bowel disease. Inflammatory bowel disease is an aberrant immune response to intestinal microbiota, and it's also a heterogeneous disease, meaning that its presentation to the clinicians and the track this disease goes through per patient differs very largely per patient, meaning it's very difficult to diagnose. But also it's a progressive disease, meaning that the inflammation over time in a patient becomes worse, and it can actually lead to flare events, or events basically where the inflammation in the intestines suddenly come up to such amounts that in the worst case scenario it requires surgery and needs a resectioning or removal of the part of the intestines. Disease is also quite prevalent. At least the last time it was counted in 2015, it was shown that 1.3% of the US adult population suffer from this disease. Meaning that it's an interesting disease to study if we can predict these flare events, because if we can predict the flare events, instead of reacting to a flare and needing to resection the colon, we could potentially proact it and give medication to suppress the flare event. So in that sense, lower the cost to the healthcare sector, but also improve the patient's quality of life, because this medication that we're going to give them to suppress the flares, while not being very pleasant medication, assumedly will give the patient a better quality of life than when they need a resectioning of the intestines. So to that end, we actually studied, we took blood from patients, 1 microliter, and we tried to see if we can identify compounds, or a biomarker as it's called, in the blood that allowed us to predict in patients that have IBD if they were going to have one of these flare events. And for that we decided to go for an interpretable model, namely a logistic regression, where we also of course took into account confounders and several other effects, but we found that we were able to have a really good marker with a very high hazard ratio that was quite convincing. And also when we went to another data set from another university, another hospital actually I should say, we were able to reproduce the findings with slightly lower numbers, but still very convincing numbers. However, when we then tried taking this marker and tried taking it to the patent attorney or to the patients or the clinicians, and we tried them to act on this prediction, we realized that explainability and interpretability is not enough, because while we understand what feature importance and feature dominance means, if you tell a patient that you want to prescribe a medication because of a certain trait, they're not really going to know. And there's this concept that is missing, which is called understandability. And it is important because it will allow you to have your audience accept your prediction in a better way. It helps you communicate to the patient because you're going to be able to use the terms that the patient is familiar with. And by doing both of these things, you will build trust with the patient. And therefore, it is important that we act on this now, and that means we need to do research into communication, but also need to take into account psychology, because a lot of these concepts that we need to do is simply not familiar to AI scientists. And storytelling and the abstraction within storytelling is going to be very important in that. Now, I hope this got your interest, and I look forward to your questions.
Welcome back. I forgot to say, today we have an extra live edition, and we had some technical difficulties going on, but we think we're all set now. And I see Bas has already arrived, so that's a good sign. So, Bas, thank you for the presentation. Could you introduce yourself briefly and Omina Technologies?
Of course. First of all, thank you for having us. As I said, I am Bas Jansen, head of life sciences at Omina Technologies. And my background is initially in biochemistry, biotechnology, you could argue. And from there, I rolled into AI and life sciences combined. And that's also the role I'm doing now in Omina Technologies, trying to take AI and applying it to healthcare and life sciences.
Nice trajectory. No wonder they put you on this one then. Now, first question, I'm just curious. I know that this is all probably fairly recent, what you've been showing, and I know you're talking about it's a process towards achieving this patent. Are there any updates? Can you tell us something about the timeline?
Yeah, it's still ongoing. There's a few hiccups, but I expect it to be done within, let's say, half a year from now, maximum.
All right, great. I hope maybe we'll see you in the conference in September, which I will be announcing at the end of today's session, to talk about this further. So the role of explainability, or the definition of explainability versus interpretability, and then on top of that, the understandability.
Yes. These are very interesting concepts and actually very important. And I also have to admit that they're not always used in the exact same wording. So how we view these things is that when you have explainability, this is the concept when you have a relatively complex model, for instance, a neural network, where you use something like Shapley values or partial dependence to give information about which feature is the one that drove a certain prediction, because it's not something you can just calculate from the model itself. So that's what we call explainability. On the other hand, you have interpretability. That is when a model is relatively simple. You can actually just calculate through the model. For instance, when you do linear regression, you don't need something more complex like Shapley values, because you can just actually calculate the prediction. Then the thing that I've gone into in the chat is understandability. Why that is important, as I said, because when you have a patient or a clinician or a GP, they do not know these terms. And also, if you try to explain it using our jargon, our language, like this feature is important, it's not going to connect to them. So you need to have this translation of what we do into their domain. And you have to take into account what is their frame of reference, their knowledge. And that's why things like knowledge graph or similar technology could be very interesting to try and bridge these two, because we need to find a way to indeed formulate what is their knowledge and what I want to tell.
Okay, thanks for expanding on it. So it sounds like the AI translator role isn't coming out of nowhere, and it's not going to go away anytime soon.
No, it's definitely going to be more and more important. And I think it's even maybe going to expand in scope. At least I hope it would expand in scope, because we have to take this much broader than that. So we need potentially people who are experts in human communications or psychology. Yeah, that domain is where I think we should think in the future.
Okay, yeah, that makes sense, because I was thinking about how are you going to describe, capture the expectations of what the knowledge is that is available within the different actors. You already mentioned something such as knowledge graphs, which would be one way of doing that. Because of course, I guess if you want to optimize for something, you ideally have a way to quantify it. So how would you see that? Is there a way to quantify understandability or to formalize this more concretely?
I do not, for now, see any way to do it objectively, but that in a way makes sense because we're talking to people, so it's going to have to be subjective. So I think the only way you can do this is to try and measure the acceptance of a prediction or how much trust, let's say, the target audience have. Now, the trust is a bit harder to measure, but let's say if in the thing I just presented, this biomarker, one is in the end the patient is being told, like, look, you're going to escalate in the disease. We can either give you a treatment that is going to short-term lower the quality of life, but should hopefully prevent such a flare event. If you can explain it in such a way that they truly accept your prediction and then are able to, let's say, not per se act on it, but make a choice on it. Like if they can tell me back, like I want the medication or I don't want the medication because, where the because is then important, I think that's the only way you can truly see if it worked.
Okay, we will come back to something like that later today as well, because what you're talking about now is also empowering basically the patients, and part of empowering is giving them more knowledge. But okay, then let's leave that topic for later today. Maybe technically, I know that you're dealing with a patent, so you can't speak openly, but could you say a bit more about how you find this, what was the innovation that you discovered?
Yes, so this disease that we talk about, there are a few ways they use to measure that beforehand. So for instance, there's this quite relatively common protein in the blood called C-reactive protein. The term is not that too relevant, but it was something that was there, and was measurable. It performed reasonably. However, over the last, let's say, 5 to 10 years, there were a lot of advances in the field of mass spectrometry, basically methods to measure smaller molecules in the blood, and also a lot of advances in the field of glycobiology, which is the modifications of the protein. So with both of these advances, it was suddenly possible to measure these things in a relatively high throughput. And then we found like, okay, well, actually, we see that these things are involved in almost all biological processes, diseases, etc. So we figured like, we have this data set, we have this consortium we're working in. Why don't we see if we can actually do something more with this data?
Okay, and so as I do understand it correctly, that this was almost an accidental discovery. When looking at the data, you realized, hey, there's more in here.
Absolutely, and this is a really good point. I am convinced there's a lot of data sets out there that have been explored initially with previous understandings, but not. They've been explored in a way like, I have a question, I answer the question, I'm done with the data, and not further explored like, is there more I can get out of this data? Now, I know this is probably, people have been saying this for years, but it stays relevant. We need to properly explore, fully explore data sets that are out there.
No, and indeed, and as you say, it's even not just the data sets that are out there. Sometimes there's also, of course, more data coming in, as you say, these new measurement techniques, the fact that data is being collected. But okay, that is a conversation that we all have heard plenty. More data, better algorithms. So let's talk about the practicals of what you are basically how you are improving people's lives. What you are doing now, is it then incorporated in monitoring and routine care? Is this an invasive technique, or what's happening?
Yeah, so when we set out to this, when we actually went, let's say, we went a bit further with the research, the goal was really to try and see, can we lower the cost of healthcare? Because when we have a patient getting very sick and we have to do resectioning, that is quite a costly and invasive procedure. So we want to see, like, can we lower the cost of healthcare, but also can we make the patient's quality of life better by using less invasive techniques? So when we can measure these things using only 1 microliter of blood that we can take once every year, because you have a quite confident prediction for, let's say, 1.5 years from the moment of measurement. But let's say we do once a year to make sure we have a bit of safety margin, a margin of safety. It's very simple. You take 1 microliter of blood, you have maybe a week in the lab, and you have a pretty accurate prediction. So it's cheap, cheaper than the current methods.
That must be a game changer, I'm sure.
Yes, and the group that I was working is still closely involved with the NHS and the University Hospital of Oxford to try and actually start using it there once we have the patent as well.
Okay, great. We'll get back to this in the Q&A. Thanks for being here. Thanks for the live interview. And I will announce the next speaker now.
Thank you for your time.
So I'll see you in a bit. We also have with us Mathias de Wachter from Ibis.ai, who will be talking about augmented intelligence for ICD-10 medical coding.
Hi, I'm Mathias de Wachter from Ibis.ai. We're an AI startup specialized in clinical language understanding. Today, I'll give you a very brief overview of a use case we're implementing at AZ Klina in Masyn, where we're using augmented intelligence for ICD-10 medical coding. It won't surprise you if I tell you that hospital funding is pretty complex. There's a definite trend towards pathology-based funding, as well as towards rewarding hospitals for being more efficient. It looks a bit like this. So a hospital has a number of beds. The medical personnel visits patients and produces records, medical information, patient files, which are then sent to medical coders inside the hospital, who read all the files for the stay and decide on a number of ICD-10 codes associated with the stay. Those are grouped together and put into statistics and then sent to the government. The government compares the statistics of the different hospitals, and based on that comparison, decides on the funding or part of the funding, which results in more or fewer beds for the hospital. There's a problem with this loop because the part where the medical information is being generated is very fast and ever getting faster. More and more information is being generated. while reading all that information and finding the correct ICD-10 codes is very time consuming. Because of that, there's a long delay, and the long feedback loop means that the hospital doesn't have the required information to make changes to improve efficiency. That's where we want to help. So our AI tool reads the medical information, and based on that offers suggestions to the medical coders who can then more efficiently code, as well as providing dashboards to the hospital. It looks a bit like this for the coders. So if you open a stay, there's a lot of information of different types, unstructured and structured. If you open, for example, a discharge letter, you can see that the most important parts are highlighted. On the bottom right, you have the list of the most confident suggestions for ICD-10 codes, and also a delta indicating the importance for the funding statistics. So when the coder selects codes, then this delta is updated. It's also interesting to note that those suggestions are based both on unstructured and structured information, for example, this lab result. So I don't have time today to show you all the capabilities of our tool, but if you want to learn more, please give me a call. Thank you.
Welcome back. And we also have with us Mathias from Ibis.ai. Mathias, would you introduce yourself briefly to the audience?
Okay, thanks. I'm Mathias Wachter, co-founder and CEO of Ibis.ai. I've been working in AI since the early 2000s, and I've started working on this problem in 2016.
Okay, thanks for being here. You already briefly talked about, or you introduced pathology-based funding in your movie. Could you give us a concrete example of what it would mean in practice for a hospital?
Yeah, so basically, it's the idea of funding a hospital stay based on the pathology instead of actions that were performed on the services delivered. So, for example, for standard things like an appendectomy or a delivery, if there are no complications, at the moment there's already pathology-based funding. It's a fixed amount for that standard thing.
Okay. And what is the relation to ICD-10 codes?
Well, so the way that these the pathologies are discovered is based on the ICD-10 codes. The ICD-10 codes are grouped together into something called DRGs, diagnostic-related groups, who that are all supposed to have approximately similar costs and length of stay.
Okay, that solves the medical jargon for now. Thanks. Let's move on to some technical aspects. Well, you're obviously using natural language understanding. What other techniques have you deployed in this solution or this program?
Yeah, so we're taking together all of the information for a hospital stay, and of course that's still largely, as I think I showed that, it's largely unstructured information, about 75 to 95% even. But there's also very interesting structured information, measurements, blood results, microbiology, and those we also include as features into our models to have a more complete view of the hospital stay.
Okay, really, one of my next questions was going to be about this combination of the lab results and the structured information. You already mentioned features. Are you using the lab results as is, or do you put extra thresholding on top of that? Because I can imagine that doctors are only interested in a lab result if it is above or below a certain threshold, something like that.
Yeah, exactly. So it depends a bit on the measurement or on the features. So for some measurements, or quite a few actually, there are medically relevant thresholds available, and of course that's what we use then because it's medically relevant. But sometimes we need to simply quantize it ourselves, and the model learns which values are above the normal range.
Okay. Yeah, you mentioned the model learns. I will come back to that in a next question. First, do you also use any time series information there, or is that too early because it's still a challenge?
Yeah, at the moment we don't use time series information. It would be very interesting to show it at least as time series when it's available, but that's on our to-do lists.
Okay, then you mentioned the model learns what, if you quantize what are interesting values or so on. I noticed when you were running the application that once a specific code was selected or highlighted, on the left side of the screen, is that the data supporting that specific code? So is there a kind of interpretability, explainability in there?
Yeah, exactly. So that's definitely one of the major features of our tool, that we have this interpretability, which is definitely necessary for anything in the medical domain. Yeah, but of course it's a stochastic relationship because we use complex statistical models, I mean machine learning models, so it's always a bit fuzzy. But
yeah, it's indeed, as you said before, statistics based indeed. But when you're talking about this kind of information coming out, I noticed in the graphical representation of how the data flows through the system that there are feedback loops or basically communication between your augmented intelligence and the medical expert or the government. But I didn't see a loop back to the medical practitioner. But couldn't this be added in the future to help them create texts that are more easily parsed, for example?
Yeah, exactly. So, and actually, I made that simplification to keep the slides easy to understand. But in hospitals, this loop is already there. So the medical coders, they are only allowed to add codes that have been verified by a doctor. But they also have other documents available, and if they see some evidence for another diagnosis or procedure, they can ask the doctor, is this relevant? This aspect, like this feedback loop, is also something that we're adding to our tool exactly in that way. So we check if, for example, in the measurements, in the blood measurements or something else, if there is something that some evidence for another diagnosis that's not mentioned by the doctor, we automatically generate a question to the doctor, and the medical coder can select it to be sent on.
Okay, so there's a kind of, that also implies that there's a kind of quality control still on the codes that are selected, because I was a bit curious about how you prominently display the delta effect of having a specific code there or not. We know that there's a bias where people start to blindly trust algorithms. Is there an ethical dilemma where you can have adverse effects where the medical coders start to click more and more ICD-10 codes to increase the funding?
Yes, that's not how the tool is devised. We don't make up codes. They're ranked in order of confidence of actually having that code. But I can imagine that let's say for a diagnosis like obesity, for some pathologies, this has an influence on the funding, and for others it doesn't. So I can imagine that the coders will click it if there is an influence, and they might not click it if there isn't, right? So it's a difficult aspect. But anyway, like we're not introducing more information with these deltas. This is something that's already available to all the hospitals.
And you have the sanity check of having the supporting information on the left hand of the screen. Anyway, a final question before we move over to the next speaker. What happens when two mutually exclusive codes are selected?
Yeah. So it can definitely happen, because sometimes similar codes are you can only choose one of the two, and they are both possible according to the model. But we also have a rule-based model monitoring the annotations, and it gives an alert
Okay. Then I said it was a final question, but I still have a tiny one. There are 60,000 ICD-10 codes. Keeping this rule base complete and up to date must be a challenge in itself. Is this handled by your application, or are you also active in this domain, or is this something for the future, perhaps?
No, like it does happen that these codes change. So every year or couple of years, it happens. It means that there can be new codes that don't have any training material available, and then we need to map it somehow. We have a lot of experience doing that from the past, going from ICD-9 to ICD-10. And of course, it's some work, but we provide that service.
All right, let's continue this conversation in the Q&A.
Yeah.
Or, well, maybe somebody else wants to have a conversation. I shouldn't be the only one. All right, no, good. Matthias, thanks again. We have from Janssen, Serge, Masyn, who will be talking about identification and implementation of disease surveillance tools for dengue hotspots around the world.
Global dengue incidence increased 30-fold in 50 years. According to the WHO, 725,000 people are killed every year by mosquito-borne diseases. There are 390 million dengue cases worldwide, and 2019 recorded the highest number of dengue cases ever. Janssen Global Public Health R&D is developing a first-in-class pan-serotype dengue antiviral small molecule for the prevention and/or treatment of dengue for vulnerable populations living in and travelers to dengue endemic areas. To support the clinical trial site selection, we aim to predict where and when dengue outbreaks will happen. This project is done in collaboration with Boston Children's Hospital, associated with Harvard Medical School, and Avia-GIS, a small to medium Flemish enterprise specialized in geospatial vector modeling. In support of site selection for the execution of clinical trials for our dengue compound, we want to identify dengue hotspots globally as far as geographical location and timing. Because of its prophylactic nature, we need enough infections to occur at the time and place of the conduct of the trial. Thereto, we want to predict outbreaks 4 weeks in advance with at least 70% accuracy, as to increase the probability of executing in a site or region with a high likelihood of dengue cases. We have first created a global vector suitability map using multi-parametric data sets, including temperature, precipitation, mobility, human access, and others. Using predictive algorithms, we now can fill the gap for locations where limited reported dengue case data exists. The performance of classical autoregression models is boosted by adding disease-related Google search term data. And to adjust for delays in dengue case reporting, nowcasting or the prediction of the present is applied. With this project, we have obtained a spatial temporal analysis of a dengue suitability map. Within selected regions, we predict with accuracies above 85% 4 to 8 weeks ahead whether the epidemic threshold will be reached. The results are combined in a dynamic dashboard showing historic dengue cases as well as forward prediction for the selected regions. The results of this work enable the development of real-time forecasting tools to support the clinical development of our dengue compound. We have achieved to reduce the risk of deploying trial sites in suboptimal locations. We will be able to provide public, private, and non-profit organizations with access to actionable information to tackle dengue. This methodology is applicable to other diseases like influenza and COVID-19. We will further continue to refine the model, achieving even greater accuracy further ahead of time in selected regions.
Welcome back. We have Serge Masyn, from Janssen with us here. Serge, thanks for your presentation. Could you introduce yourself briefly to the audience?
Yes, hello. My name is Serge, Serge Masyn. I'm a data science lead for global public health research and development at Janssen. Computer scientist by training. Started off in speech recognition 30 years ago with a well-known Flemish company. Followed a classical IT trajectory, and then 5 years ago came back to my first love, data science, machine learning, and global public health.
Glad to have you back here. Could you say a few more words about this global vector suitability model first to understand the context of the conversation? What kind of dimensions are you taking into account there?
Okay, so one of the challenges with dengue is that it is often misclassified as malaria because it has similar symptoms, and it's often underreported, especially in Africa. So what we're doing here, we're taking parameters such as temperature, rainfall into account, as these are good predictors for the vector, for the mosquito. Where do they breed? Where do they live? On the other hand, you need also humans to be infected, and so elements like human accessibility, poverty, urban or rural setting are also good predictors for the combination of having both vectors and humans to have dengue cases. So with known dengue cases and with these parameters, we create a predictive model to cover like the whole map of the world to identify the risk of having a suitable place for dengue cases. That's the vector suitability map.
Okay.
Yeah.
Thanks. And you're saying to predict, do I understand correctly that you are able to do this up to a year in advance, or?
No, not really. So the vector suitability map per se is broader, shows you endemic areas, if you like, where there's a lot of dengue cases. On the other hand, we also need the temporal aspect. And while setting up a clinical trial takes about a year time to do so, it is very difficult to predict really from a temporal perspective so long in advance. And therefore, for that, we are using Google search term frequencies in combination with classical epidemiological modeling to predict 4 to 8 weeks in advance whether an outbreak will occur at a given location, yes or no.
All right, so is this, you say you're using Google searches as well, is it the global vector suitability model then coupled with another model to?
Yeah.
Okay.
Yes, indeed. It actually covers the entire span because looking at where the vector, the mosquito can live and breed is early in the process. You need them to bite people and then pass on the infection. But the Google search frequencies are actually at the end of the process when people start to experience symptoms like vomiting or heavy bone pain. And so in combining actually the life and death of the mosquito together with what people experience, we hope to cover a broader span in our ability to predict where things will occur.
Okay, yeah. Things occur, outbreaks, you're talking about accuracies. What exactly are you quantifying when you're talking about an accuracy? Are you talking about the size of the outbreak or the timing of the outbreak?
We are trying to predict whether an epidemic threshold is going to be reached. Today we are a bit more familiar with that. So epidemic threshold is the number of cases per 100,000. For example, 300 people being infected on 100,000. And we need, unfortunately, we need infections to prove that our compound is working. So we are predicting whether we are surpassing that threshold, yes or no. That's one element, because that's an indication that there will be enough infections to conduct our clinical trial. On the other hand, we have the temporal aspect. We want to assure that we conduct the trial in a location where there's going to be enough, at the time where there's going to be enough infection. So we try to do that 4 to 8 weeks in advance so that we can adjust, say, the resourcing, the staffing of the clinical site, whether there's more cases or less cases. But we don't need the exact number of cases. We just need to know enough or not enough.
Okay, so you could then reshuffle your resources to the top case sites, for example? So make a ranking of the sites rather than having exact.
Our first prediction will give us a very broad view of where we need to be. At the end of the day, we'll have, say, a top 10. And with the temporal aspect, 4 to 8 weeks in advance, amongst the top 10, we will say, you number 3, number 5, number 7, spin up because we are expecting an outbreak, and the others stay put.
Okay. If I could go a little bit more technical, because I'm still a bit confused that the societal definition of an epidemic or a threshold is also the most relevant one for a trial site suitability. How should I put this? You are making a binary choice or making a binary prediction, but behind the scenes or behind the machine, maybe even, there will be numbers that are being predicted. The size of the outbreak will be somewhere in this model or the probability. Do you ever use that kind of information as well before binarizing it?
Well, it is of course the case that the trial design sets a certain threshold for the number of patients that would be required. And where I say the epidemic threshold is reached, you could of course translate the number of patients that is being required as your threshold to predict above or below that certain threshold. So it doesn't have to be the number that was put forward by a Ministry of Health or a government. It can also be the output of the trial design in numbers of patients.
Okay, thanks. That answers my question.
Yeah, but typically when you predict the number of cases, somebody makes a binary decision afterwards in their head.
Okay. You talk about governments and policies. That brings me to an ethical consideration or an ethical question as well. I understand that for your compounds, as you rightly mentioned, unfortunately, you need afflicted people. Mm-hmm. At the same time, when you know that that there's a high probability that there's an outbreak coming, local policies could do something to curb the spread of the mosquito or so on. So how do you balance those two, global responsibilities, you'd say, long term and the short term?
Exactly. We are global public health as a department, and so it's high in our standards and in our mission that this type of modelings, we are going to put that into the open so that the global public, if you like, or the targeted areas can benefit from the results of this modelling. So to that extent, when it becomes available, ministries, governments will be alerted, hopefully 8 weeks in advance, that an outbreak is coming, and they would be able to take proactive measures.
Okay, that's great news. Thank you, Serge. We'll see you back for the Q&A.
Okay, sure. Thank you. Bye.
Our next speaker is Annemie Ribbens from Icometrix. She will be talking about towards value-based care of neurological disorders.
Towards value-based care of neurological disorders. 1 in 6 people worldwide are suffering from a neurological disorder. Brain MRI scans play a crucial role in terms of detection and monitoring, but less than 10% of the scans is actually acquired using an optimal protocol, and radiological reports often show discrepancies. Additionally, the brain MRI scans are insufficiently used in the decision-making process, which still majorly relies on the clinical assessment. And this clinical assessment can vary a lot between neurologists. Finally, there is an increasing empowerment of the patient, and this is great. However, patients are often insufficiently informed about the disorder and fail to report symptoms to their physician. As such, this all results in a large variability in the treatment decisions between neurologists, insufficient detection of the actual disease course, and patients with a poor adherence to their treatment. So in order to provide the most optimal treatment to each patient at the right time, reliable monitoring tools are needed to assist providers and patients. In this context, we need monitoring tools that are reliable quantitative and observer independent. Additionally, healthcare providers should be informed about treatment guidelines, and they can benefit from tools that guide decision making. All these tools should of course be well integrated in the clinical workflow. Icometrix developed AI and deep learning based solutions to improve the patient's care pathway. In particular, we developed the IcoBrain imaging solution, which is an AI-based software for quantitative analysis of brain MRI scans in terms of lesion volume and whole brain volume. iCompanion, which is a patient app to track symptoms. iCompass, a set of AI-based tools that guide clinical decision-making. And all these tools are optimally integrated in the clinical workflow. Finally, all these aspects are gathered together and the data is gathered in our iContainer platform. This platform of solutions is fully regulatory cleared and already rolled out in hospitals worldwide. Additionally, we collaborate with patient organizations, registries, and provide these solutions to top pharmaceutical companies. If you like more information, feel free to contact me or visit our website.
Welcome back. Welcome, Annemie. Could you briefly introduce yourself to the audience, please? Thanks for being here.
Sure. I'm Annemie Ribbens. I'm the VP Clinical Trials and Evidence at Icometrix, and I'm responsible for bringing the Icometrix solutions towards clinical trials for optimal assessment of drug efficacy, as well as to generate evidence of using our solutions in clinical practice.
Okay, clear. You were talking a lot about the why and the what. Could you tell us a bit more about the how, some keywords, some techniques that you are using?
Yes, definitely. So as you noticed already a little bit in the movie as well, is that one of our solutions is the IcoBrain product. This is a quantitative tool, so an AI-based and deep learning-based tool that quantifies brain MRI scans. Excuse me. So it quantifies basically the whole brain volume as well as lesions in the brain, for instance. For instance, in multiple sclerosis as such, you know the exact values of lesions, the number of lesions, also how much your brain is shrinking, and you know exact values of that, instead of, if you look at a radiological report nowadays, they are still majorly descriptive. So they're just majorly saying like, I see there might be a new lesion, there might be some volume shrinking, but you don't know how much. So this is one of our solutions. And a second one of our solutions is our patient app. So if a patient comes to the doctor normally, they have 15 minutes with their neurologist, for instance, where they can talk and they can describe their symptoms, while patient app allows to track during the year how the patient is doing.
And how does it do that? Does it prompt the patient randomly, a fixed time set, or is it linked to a wearable?
It's really a patient app on your phone, and you're free to fill out symptoms whenever you like. But there are also reminders included. We have some educational content to stimulate patients to fill this out. But in general, we notice, for instance, we have rolled out this app in multiple sclerosis. We notice that people are really involved with their disease and are really actively using and have a good adherence to the patient app.
Okay. A few questions about the numbers in your slides. Some of them are a bit frightening or disconcerting. Less than 10% of the scans adhere to the quality standards, 24% variability. Is this unique to this problem? Is this common in medical imaging?
It's a very good question. I think that the 10%, for instance, that really relies on brain MRI scans in multiple sclerosis, there the community has done a lot of effort to find like what is our optimal protocols. So for instance, you have the CMSC guidelines, the MACNIMS guidelines, but we see indeed that the practical implementation of these guidelines in clinical practice is really lacking. So it's indeed less than 10% that's actually adopting these guidelines. The 24% of radiological error is in the neurological space. And again, I think brain MRI is even one of the areas where people have invested a lot on what is optimal and have provided guidelines. I think if you go beyond that, of course, I'm not very familiar with other fields, but I think if you go beyond that, there might be even larger variabilities.
Okay, then a last question about the numbers, talking about the variabilities. You also mentioned this 30% error rate. Is it then due to this variability, or where is this coming from?
Yeah, the 30% error rate is really actually the difference between neurologists. And one of the measurements, how this is quantified, so it's rather on the clinical assessment than on the brain MRI scans. And one of these numbers where it's coming from is, for instance, again, in multiple sclerosis, they're using a disability scoring system, which is called the EDSS. And if they address the EDSS by 2 neurologists, we know that's quite subjective. It relies, for instance, also on what patients are reporting in terms of symptoms, and we see that there can be a difference of around 30% in these scoring systems.
Okay, so the 30% is an indicative of disagreement between experts. Does it then mean that there is no real ground truth as such?
Yeah, that's a very good question. I think, yeah, everything, and we also heard it in the previous talk, it's very difficult to do prediction, but also already to do assessments, because a lot of aspects, definitely in terms of clinical symptoms, and if you try to put a scoring based on that, it can be quite subjective. So we really have to train raters to act in a similar way and decrease your inter and intra-rater variability. But these scores are used in a lot of clinical trials. So although there is some variability, they're definitely useful. They have shown to be like reasonably good endpoints to validate, for instance, drug efficacy.
Okay. As in this context where experts disagree, I find a fascinating way to tackle this is to, rather than predicting labels as such, to talk about the probabilities that the label would be assigned. But that would be a discussion for some other time, perhaps. You talked already about your patient app and how it explains things to the people. You mentioned patient empowerment as well. We already talked about explainability and understandability. So how does your app help make things understandable to patients?
Yeah, that's one of the key points, and we have indeed an educational center also in the app. There you can find basic questions concerning the disease. We know, for instance, that a lot of patients are not really aware about their disease, and definitely not about the disease course, which we also heard in previous talk. But indeed, like in terms of understandability, what we're doing is we work a lot together with patients through patients' organizations, but also directly doing user experience studies and so on.
Okay, because I was going to ask, do you have an idea about the real-life impact of what this means for patients' lives? But through user studies, I guess that's what you find out there. Okay.
And yeah, the app is also, yeah, it's released last year. We have until now a very good adherence, but of course it's still, everything is still in the rollout phase at this point in time.
The app helps in tracking long-term evolution, which is of symptoms, which is always difficult for both medical practitioners and patients alike. Does it already do a kind of modeling or smoothing as well, or does it derive summaries from it?
Yeah, again, good question. So what we're doing is basically we have a healthcare practitioner portal, which is really integrated in the clinical workflow. And what you find in this healthcare practitioner portal is, first of all, the results from the quantitative assessment of the brain MRI scans, so where you have the volumetric measurements. Then on top of that, you get the clinical scores and the symptom trackers that you got from the app. And there you get general overviews of how they are evolving over time, when they have addressed it and how it is looking. And then finally, we developed Icometrix, which is a set of predictive tools. And based on all this information from both the brain MRI scans as well as from the clinical symptoms and the clinical information, we do an estimate of how the patient might evolve over time, which help then guiding the neurologist in their decision making. It's not a decision tool; it's a tool to help the neurologist guide in what we expect in how the patient will progress in the future.
All right, so you're not putting the neurologists out of business just yet.
I think that they will always be necessary because there are things that you cannot capture, which is just like a human factor in there. So, but it's help guiding. The big advantage of AI is basically bringing a lot of data together and seeing quick resemblance between patients from all over the world, for instance, instead of just from your core lab and your own experience. On the other hand, these kind of extrapolations, I think they're kind of own to the or seeing adverse events, things like that are uncommon. These are really human factors, and also, of course, the personal relationship, things like that stay important.
What we often say is to provide medical practitioners with the tools to base themselves on the experience of all of medical history and all of their colleagues around the world. So yeah, but when you're talking about around the world or services together with hospitals, how do you integrate that with other tools? Because you are not the only ones out there. Does a FHIR standard play a role there, or is it something else entirely?
It's a very good question. Icometrix does indeed a lot of effort to integrate to the best as possible into the clinical workflow, as well as different platforms as well. We have a very interoperable platform, we have already, for instance, a fixed integration with PACS systems, so images are directly sent from the hospital to the Icometrix cloud and vice versa. The results are pushed back into the cloud. Of course, these data standards like FHIR, they help to further improve and help us push AI solutions better into the clinical workflow and vice versa, to also pull information from there. So it's definitely one of the key aspects. Unfortunately, FHIR is only part of version 7 of HLL7. Most hospitals are currently at version 2, so still some time to wait.
Okay. We're going to wrap this up for now. We're going to return briefly to Bas as well, but we'll be seeing you at the Q&A. Thanks again for being here.
Thank you.
All right, for the people who joined a bit later, our first talk of today encountered some technical issues, so we'll be reprising the little presentation that Bas from Omina Technologies provided us with, and we'll be having a talk afterwards as well. So now we'll have extending explainability to understandability, an IBD escalation biomarker study by Bas Jansen from Omina Technologies.
Good day, everyone. My name is Bas Jansen. I'm the head of life sciences at Omina Technologies, and today I want to talk to you about inflammatory bowel disease. Inflammatory bowel disease is an aberrant immune response to intestinal microbiota, and it's also a heterogeneous disease, meaning that its presentation to the clinicians and the track this disease goes through per patient differs very largely per patient, meaning it's very difficult to diagnose. But also it's a progressive disease, meaning that the inflammation over time in a patient becomes worse, and it can actually lead to flare events, or events basically where the inflammation in the intestines suddenly come up to such amounts that in the worst case scenario it requires surgery and needs a resectioning or removal of the part of the intestines. Disease is also quite prevalent. At least the last time it was counted in 2015, it was shown that 1.3% of the US adult population suffer from this disease. Meaning that it's an interesting disease to study if we can predict these flare events, because if we can predict the flare events, instead of reacting to a flare and needing to resection the colon, we could potentially proact it and give medication to suppress the flare event. So in that sense, lower the cost to the healthcare sector, but also improve the patient's quality of life, because this medication that we're going to give them to suppress the flares, while not being very pleasant medication, assumedly will give the patient a better quality of life than when they need a resectioning of the intestines. So to that end, we actually studied, we took blood from patients, 1 microliter, and we tried to see if we can identify compounds, or a biomarker as it's called, in the blood that allowed us to predict in patients that have IBD if they were going to have one of these flare events. And for that we decided to go for an interpretable model, namely a logistic regression, where we also of course took into account confounders and several other effects, but we found that we were able to have a really good marker with a very high hazard ratio that was quite convincing. And also when we went to another data set from another university, another hospital actually I should say, we are able to reproduce the findings with slightly lower numbers, but still very convincing numbers. However, when we then try taking this marker and try taking it to the patent attorney or to the patients or the clinicians, and we try them to act on this prediction, we realize that explainability and interpretability is not enough, because while we understand what feature importance and feature dominance means, if you tell a patient that you want to prescribe a medication because of a certain trait, they're not really going to know. And there's this concept that is missing, which is called understandability. And it is important because it will allow you to have your audience accept your prediction in a better way. It helps you communicate to the patient because you're going to be able to use the terms that the patient is familiar with. And by doing both of these things, you will build trust with the patient. And therefore, it is important that we act on this now. And that means we need to do research into communication, but also need to take into account psychology, because a lot of these concepts that we need to do is simply not familiar to AI scientists. And storytelling and the abstraction within storytelling is going to be very important in that. Now, I hope this got your interest, and I look forward to your questions.
Welcome back, Bas, and the rest of the audience. Thanks again, Bas, for being here for the second time. Would you introduce yourself and Omina Technologies briefly?
Of course. First of all, I would like to thank you for having us. And my name is Bas Jansen. I'm the head of life sciences at Omina Technologies. My own background is more in biochemistry and informatics, and from there I rolled into AI, trying to use this during my previous life, let's say. And in my current role, that is taking AI and trying to apply it to actual the life science industry, the healthcare industry, but in a way that is actually useful for patients as well, rather than this vague thing that doesn't tell them anything.
Yeah, I can imagine it gives you a real drive to deliver your best if you're having an actual impact in patients' life, and in the tool that or the solution, the innovation that you've described just now. I know that you're still in the process, and it hasn't been that long since you sent us the video. Do you have any updates, or do you have a timeline on when this patent will come out or be accepted?
We have a rough estimate. For now, they're still at the patent attorney in the UK, and they're also trying to do it in the US at the same time, because there's a small difference there. I'm not going to pretend I know the legal implications of it, but it should be within half a year, last I was told.
Okay. Well, yeah, let's maybe have an update for us on the AI4growth conference later this year in September.
Mm-hmm.
Today's theme is has been explainability and interpretability as well, even understandability. Could you share us your definitions of those and how you came to understand the importance of them?
I think it's interesting that you indeed say our definitions, because we had quite a few chats already, and it is clear that there's no clear definition of it. I mean, it differs a bit. The interpretation we have been going with is that explainability is something that you can provide for a model that is quite complex, like a neural network, by using a concept like a Shapley value or a partial dependence plot to tell you which feature drives a prediction. For instance, in a neural net, they could say that if you have feature X at a high level, this is probably what causes your prediction. Mm-hmm. That's explainability. On the other hand, then we have interpretability, which is something that a relatively simple model is by definition interpretable, because it allows you to literally calculate or basically do the calculations that the model did itself. For instance, linear regression. However, of course, both of these concepts are concepts that we know as AI scientists. If I go to a GP or God forbid even the patients, and I need to explain why I made a prediction about him, things like feature importance are not going to tell these people anything. So you have to translate the prediction. Let's say prediction, because you could argue you may need to translate the model as well, but let's go with just a prediction to the patient. Like, what does this mean? How did we determine this? In a way that they understand. So you have to try and translate the knowledge of your model and your prediction into a dictionary or language construct that they understand. So you need to, in a sense, almost find a formal definition of their knowledge reference and then match this with the model, where indeed recent concepts like knowledge graphs can be useful.
Now that I hear you explain, is it then that basically explainability is for black box models, interpretability is for white box models, or?
That's how I have been working with it, yes.
Okay. Okay. Yeah. All right. Yeah, makes sense.
Doesn't mean that things like Shapley can also be, of course, be applied to white box models, but you could argue why.
Yeah, indeed. But this conversation also, especially when it comes to understandability, brings us to the AI translator role that's been upcoming the last couple of years. Isn't going to go away anytime soon, clearly.
No, I think it's going to be more prevalent than ever. Now that AI, in a sense, is maturing, and when I say maturing, I mean because we are thinking about these concepts, explainability, interpretability, understandability, means that there is going to be a bigger need for this role of AI translator. And I think it's also going to expand in scope, because you're going to need understanding of psychology, human communication, and also how to define knowledge in an abstract way that, yeah, it is just something that most AI scientists that I know are unable of doing.
You need to eliciting the frame of reference of your patient or your non-expert, capturing that formally is a huge challenge, much less trying to quantify your understandability to track progress. Any thoughts there, how to tackle that problem?
It's interesting because, of course, when we talk about quantification, we usually think about numbers, like actual numbers you can measure. But, of course, understandability is by definition a subjective matter. So I don't know of a clear way of quantifying it, but I think at least what to me would be indicative that it worked, that if we take the research we just did, if we have a prediction, we go towards a patient and we say to the patient, like, look, because of our prediction, let us assume, well, let us hope they understood it, we think you're going to have a flare event. So you can choose to take the medication that will impact your quality of life in the short term, making it slightly less, but you should not have, hopefully, a flare event. If this patient is then able to answer, like, I will take the medication or will not take the medication because, that to me would indicate that they understood it, accepted it, and we are able to communicate about it. And hopefully that way they will trust the prediction. Okay,
this also brings back the concept of patient empowerment from Annemie's talks. Yeah? It's all coming together nicely. Great. When you're talking about patients and collecting data, could you say a bit more about how the experiment was set up, data collection, a historical or design experiments for this purpose?
Yeah, this research was actually done in FP7 consortium called the IBD Biome Consortium, which was several, I believe it was the University of Leiden, Oxford, Magdeburg, and there's a fourth one, but it skips my mind right now. But in that consortium, they took patients with IBD, collected blood, and tried measuring them across various institutes in Europe. And the institute I was working at that moment used a sample set that was previously published by another university to basically prove their own method. And when they had measured this, and when the initial investigator was looking at this data, I realized that there's more in here. There's this entire time domain that nobody's looking at. And that got this ball rolling. And in the end, it was like, why don't we actually, instead of just trying to see if a patient has IBD, why don't we do something with the escalation or progress, which no one had done before? So indeed, the data was there. And I think the key thing is people collect data for a question, but then they kind of forget to explore it further.
No. Does this has things in common with real world evidence-based research then, as far as I understand? It wasn't entirely hypothesis-free modeling, right? Because you were looking for markers to predict a flare event in a person suffering from IBD.
Partially. Like the data set was collected initially for patients with and without IBD. So the initial analysis was on trying to identify if a patient has IBD, because to identify IBD, sorry, is inflammatory bowel disease, if people have forgotten it, but to identify it, we usually look at certain proteins in the blood. But the old method was not very accurate. And with the advances in the recent fields of glycomics, which are basically small modifications of protein, as well as new experimental techniques, suddenly it is possible to measure these things. So within the consortium, they first looked, can we stratify or identify patients with IBD first with a higher accuracy? But when they were collecting the data, they also took into account people who have IBD already. How did they progress? And what progress here means is over the time, do they get more severe medication? Do they have resectioning events, which is where they remove part of the colon? That made me think, like, if we have this and we have this glycosylation data, which is involved in almost any biological process, can we actually predict such an event from happening before it happens? And in that way, lower the cost to healthcare significantly, because if you don't have to go into surgery and just are able to measure this from 1 microliter of blood, maybe measured or taken once a year, that's very non-invasive and very relatively cheap compared to surgery.
And indeed, and of course, the patient's quality of life. So you're not, okay, so it could even be a early way to screen for IBD. So rather than just trying to find first flare.
In theory, yes, but the data to identify patients who are going to develop IBD was not convincing enough for me. Maybe the sample set was not large enough. I cannot say. The only thing I can say for certain is that within patients that already have IBD, we can predict such a flare event from happening.
Okay, but it does sound like when there's probably the markers that you identify that now turn out to be important for IBD or flare events, there'll be similar or other markers for other diseases out there. Are you still active?
Oh, yes. Yes, not with that group anymore, but in my current role, I am trying to collaborate with quite some research group because indeed, if we look at the field of glycosylation, it is involved in almost every disease, especially autoimmune diseases. And of course, it's very hard, almost impossible to prove if it's a cause or effect, but you can measure it. It's very easy to measure and it's very accurate.
No? Fascinating story. Thanks again.
Thank you for your time and thank you for having me as well.
This concludes the interviews for today. Before moving on to the Q&A session, where we hope to hear from the audience as well, and from the speakers among each other even, I would also like to briefly talk about the AI4growth conference later this year. It will be held on September 16th. We've called it AI Your Business. Visit us on the website, register to receive updates, and we hope to see you there. It's going to be a full day event. Now we're going to take a 2-minute break before we start the Q&A. So see you soon. All right, welcome back to the live Q&A. We now have all the speakers on the screen. If you would like to ask one of them a question, I propose you raise your hand using the Microsoft Teams tools that are there. Unmute yourself once your name is called, and obviously ask your question. If there's not an immediate question, I would then ask to Mathias a bit more about the state of Dutch language understanding, and especially in this medical context, because I can imagine it must be an absolute, not nightmare, but a challenge.
Yes, so in English there are different biomedical pre-trained models available, because of course there's a lot of publications in English, and it's easy to find medical data in English. In Dutch, I'm not aware of such things, or no big volumes anyway. So of course it's very important for us to partner with hospitals so we can use their data to train models.
And using their data is probably, it takes quite a lot of preparation and anonymization and all that kind of stuff.
Yeah, of course, there's especially anonymization is very important, and there's a lot of focus on that, and of course general security and
yeah. And therefore for Annemie, because you're also dealing with patient data, how have you handled that problem in the training phase and during the deployment?
Yeah, all the data that we receive are fully anonymized. And in the patient app, for instance, where they give patient information, they also just sign an informed consent form for what the data is used, of course. So we have also data agreements, of course, with all the hospitals. Further, Icometrix is ISO 27001 certified for information security. And we also comply, of course, with aspects like the GDPR, privacy acts, and so on.
Okay. Bas, you had a question. Yes.
Actually, I think this is an interesting point, because indeed also in our study we had anonymized the data, but the problem is with a rare disease like this is it's not possible. If you tell me a patient's descriptors, I will be able to tell you who the patient is, because they're that rare. So I think this is a really interesting discussion that should be explored really properly by the let's say the powers that be. How far do you go with this, or how useful is it? Because on the other hand, there may be certain identifiers that are considered protected, that could be very important for the diagnosis.
Mm-hmm. Yeah, I actually fully agree. And we have quite a lot of these discussions at hospitals as well. We're receiving, for instance, brain MRI scans, and most of the hospitals, they're quite fine with it. But for instance, if you do a 3D reconstruction, you can recognize the face quite well. And now and more people are doing, for instance, defacing, so that you cannot reconstruct the face anymore. But then in the end, you can wonder, like, the brain might be far more unique actually than the face itself. So how far do you go with it? So I fully agree. It's a difficult discussion, and I think we're not yet at the end of that.
I think it's going to even go further and further all the time. If we look at omics or multi-omics, which is, I have to admit, my first love, the more dimensions of data we get of a patient, for instance, genes, glycans, proteins, etc., one would imagine, at least I would imagine, that once you have all the dimensions available, that everyone is unique. It's like a fingerprint of all your data. So it is, in the end, completely identifiable.
And it's, well, you were able to basically track somebody or notice somebody throughout the system at different points. Indeed, even the fingerprint of the brain, for example. But that is something that you need. You need to know which measurements belong to which patient. There's a difference between getting them personally identifiable, of course. But we also see in anonymization that it is not a binary thing. We have this K anonymity and so on. There's a lot of work or attention there as well, and rightly so. So how do you approach well, does one of you have experience with the different degrees of anonymization?
The minimum anonymization, I think, that can be done, and as far as I have seen, does not hinder the use of the data, is just by removing, for instance, address. Well, actually, address is already questionable, but name. That one can be removed. All the other features you can question, does it not have actual importance, because someone's address has an impact on where they live, environmental variables, etc. So,
yeah, I also agree. I think what is also quite surprising is that most of the time these instances on privacy have a lot more issues with what's going on than actually the patients themselves. So patients, they're really involved in the disease, and they want to move the disease and the cures for the disease forward. And they're actually quite happy to share their information, as long, of course, that they know that it is used in appropriate ways. That's of course something that you have to be careful about. But in that sense, patients are quite involved and quite happy to share their information.
Indeed, but that's also a double-edged sword, right? We're balancing the short term, the needs of myself, with the needs of society at large. You say, if you have nothing to hide, then why do you want anonymization? Things like that. But okay, that's a whole different discussion. But thanks for your perspectives. A little thing about maybe genotypical diversity, or the like global diversity. I see this also coming up in, for example, with Serge, where you need to have different population groups can respond to disease in different ways. And Annemie, culturally, communication is different in different groups, with Bas as well, the explainability aspect there, and the same even for Mathias. So maybe let's start with Serge to keep it simple there. Your dengue is of course very constrained in dengue hotspots. So it's normal that you won't set up a dengue trial in the north of Sweden, for example. But this diversity, is this receiving enough attention in your view with global span?
Well, as far as the vector is concerned, no. But from a human perspective, indeed, for each and every location, we look into the Google search term frequencies that are relevant. In another study we have seen, or another work, we have seen that parts of the world, cultures respond differently. So not only people react differently to a disease, but cultures respond differently on social media, by looking through Google to certain aspects of the disease, and therefore in particular areas of the world, people will look for this and this and that, and others they will look for others. So the model needs to be adapted, if you like, to those locations. There is no such thing in this case as a one-size-fits-all model. Not from a human perspective, from human behavior perspective, and also not from the parameters perspective. Seasonal aspects on the coast of Brazil are not at all seen, say, in a city setting of Singapore, which is highly endemic. The middle of Latin America, Colombia, Medellin, sees the impact of El Nino totally different than the coastal areas. So this model is not a one-size-fits-all model. It is like a strategy and methodology. If it works for another location, fine, but you will need to take the local perspectives, both vector-wise, mosquito-wise, and human-wise into account.
For sure, indeed. And you mentioned updating your model. We haven't talked about climate change today, but that will also impact your dengue mosquitoes. What you mentioned about the Google searches, but updatability of model reminds me of an interesting anecdote as well, where Google used to do flu predictions based on the searches, until they started basically adapting their search suggestions once people started typing. So they were basically interfering with their own data collection, that suddenly the flu symptoms were being found more often just because they were suggesting them to people to look for them. So that kind of unexpected side effects can be quite head scratchers. Matthias, have you seen any unexpected outcome of deploying your solution at hospitals, that they realize that, hang on, something is off here, something weird is happening?
Well, the fact that there's definitely also a lot of diversity. We can't just take one model from a hospital and use it in another hospital. So there are dialects, let's say, per hospital, both in the language that doctors use, also the information that is actually stored and available to us. That's also important, and even the way medical codes, which guidelines are used internally, because there is some freedom. So yeah, for sure, we see a lot of variability as well.
All right. Do we have another question from the audience? Yes, Philippe, just on cue. Please go ahead.
Okay, yes, hello. So my name is Philippe Leroux. I'm an innovation officer in AI at Ghent University. First of all, I would thank you all for the very interesting presentations. But I have one specific question to Annemie. So Annemie, as far as I understood, so at Icometrix, you have designed very, you're covering a lot of different pathologies which relate to the brain. Now my question is, how far are the models that detect the abnormal evolution of a specific pathology? How far are these tailored towards the pathology? And do you also have a solution, or do you think it's even possible to have a solution that is also tailored, that is kind of a generic model to detect abnormal patterns within the brain for patients that are not yet diagnosed with a specific pathology?
Yeah, very good question. So first of all, we tailor indeed most of our solutions towards a specific pathology. So all the tools that we brought to the market are specifically for disorders. So for instance, at this point in time, we have a product for multiple sclerosis, a product for dementia, more specifically for Alzheimer's disease. We have a product for traumatic brain injury, but even those actually capture a lot of subgroups as well. So you can tailor and tailor. That's one aspect. And the tailoring is sometimes quite important because you look for very specific features. You want to classify those according to very specific settings. But on the other hand, there are indeed also tools on the market, and also Icometrix is working on those to just detect if something is abnormal in the brain. So we have, for instance, published one deep learning based method where we just detect like, okay, a normal brain would look like this, and abnormal, and this is the brain that we received. So these are the parts in the brain that are actually abnormal. So we advise, in that case, to a radiologist to look at these particular parts. These are some of the research studies that we are currently doing. We haven't brought this to the market yet, but indeed that's definitely also an angle that we are investigating.
Okay, thank you very much. That would be very interesting. I'm looking forward to that. Thank you.
All right. Do we have time for one more question from the audience or from the speakers? Otherwise, we will wrap it up here. Then I would like to thank the speakers once again for their time, flexibility, and good cheer today, and as well as their interesting talks, of course. I hope to see you in a real-life event soon. Maybe, again, plugging it really hard here in September at our AI4growth conference, but before the time as well, of course. Yeah, if somebody from the audience would like to get in touch with one of the speakers and can't find their contact information, of course, reach out to us first and we will happily put you in touch. And that's it for today's session of AI4growth. So I wish you all a pleasant continuation of your afternoon, evening, and see you next time.